Hi, I'm Carly,
And I'm Erin.
and in our project, we studied apigenin's ability to offset abnormal regeneration induced by upregulation of the WNT pathway in planaria. Cancer, the uncontrolled growth of abnormal cells, is the second leading cause of death in the US. However, current methods of treating diagnosed cancer are not always effective and can have serious side effects. The WNT pathway is an important biochemical signaling pathway. Its abnormal upregulation has been associated with several types of cancers and their processes, such as tumorigenesis, angiogenesis, and cell migration. Thus, it is important to focus on the WNT pathway in order to improve and contribute to existing treatments for cancer.
In normal cells, the WNT pathway is involved in important cellular processes, including proliferation, differentiation, and migration. During the upregulation of WNT, beta-catenin, which is involved in migration, is upregulated, and GSK3 beta, which is involved in proliferation, is downregulated. In multiple myeloma plasma cells, lithium chloride has been shown to upregulate the WNT pathway, and this was observed through increases in beta-catenin and decreases in GSK3 beta activity, which ultimately caused decreased cell survival and apoptosis. Lithium ions also cause malfunctions in body patterning and colonial chordates, causing changes in cell shapes. Apigenin has been shown to inhibit the activated WNT pathway in cell studies, including colorectal cancer cells, cancer stem cells, and osteosarcoma cells. Apigenin also impeded the growth of colorectal cancer cells and cancer stem cells up to 90%.
Planaria are regenerative flatworms with neurons and physiology that are similar to humans. The WNT pathway is also involved in planarian regeneration in the head versus tail polarity decision. When WNT signaling is low, eye spots develop to form heads. When it is high, tails are formed. For our goal, we strive to examine the ability of apicnin to be an effective treatment for abnormal regeneration that was caused by the upregulation of WNT through LICL. We strive to use apicnin as a treatment for abnormal planaria by inhibiting the WNT pathway. We decided to use planaria because it is important to be able to observe the effects of apicnin on the WNT pathway in a model organism rather than simply cultured cells. We hypothesized that apicnin would be able to mitigate the effects of LICL with concentrations at or above 10 micromolar being the most effective. All planaria were cut at the beginning of each trial. We conducted 5 total trials. For our experimental setup, each group had 6 head and 6 tail fragments. Our positive control group remained in a salt solution for the entire trial. We had 3 experimental groups that were placed in 2 millimolar LICL for 2 days. Afterwards, planaria were transferred to their respective solutions of 1% PEG, 10 micromolar apicinin, and 20 micromolar apicinin diluted in 1% PEG. Since apicinin needed to be dissolved in a non-toxic PEG solution, we used the LICL2 PEG group as our negative control. Regarding our assays, we carefully monitored planarian regeneration for regeneration landmarks over the course of 10 to 15 days. The PLMV assay measured the speed of head fragments of planaria by measuring the distance traveled in 1 minute. Slower velocities indicated hindered brain activity. For the odd shapes assay, planaria were observed under a microscope for abnormal shapes such as C shape, screw shape, and other contractions. These shapes were indicative of toxicity. For the light-dark assay, planaria were placed in the light side of a petri dish. The number of planaria that crossed over to the dark side were counted. Since planaria have a natural preference for darkness, staying in the light was seen as abnormal.
On this slide, we can see the different stages of regeneration of tail fragments. We used a rating system on a scale of -2 to 7 to score regeneration stages. Key benchmarks included death of planaria, wound healing, eye spots forming, and the head shape becoming more distinct. These line graphs show the average regeneration of 3 tail fragments in each of 2 trials which were conducted at school. We also conducted an extra third trial which exhibited similar trends. All LICL groups initially lagged 1 stage behind the control, but the 20 micromolar picketing group caught up to the control by day 6 in trial 1 and day 14 in trial 2.
These results show regeneration progress of planaria in a third official trial. Since it was done in a separate location, the conditions varied slightly. We did do extra trials and they showed the same trends. We had modified parameters for this trial. -2 was used as an indicator for dead planaria, which were first observed at day 3. In this trial, all the planaria and the LICL to PEG group died. The overall trend that apicnin restores LICL hindered regeneration was reinforced. Overall, both the apicnin groups lagged behind the control group, but 20 micromolar planaria regenerated slightly faster than 10 micromolar, and both were markedly better than the LICL group that was not treated, in which all worms died. These images show progressive regeneration stages observed in trial 2. Here are some images that give a qualitative sense of what we were measuring.
This clustered bar graph shows the PLMV assay. The control group shows a small upward trend except for day 0, and for all LICL groups, locomotion increased substantially over time. However, the two oppigenin groups displayed decreases in velocity after day 8, possibly indicating that continued downregulation of WNT could have some negative effects. PLMV and other repeated trials showed similar results. The control group's velocity in the other trials was as constant as in this trial. However, in other trials done at home, the 20 micromolar and 10 micromolar opinion groups did show decreased velocities over a longer period of time, although not as dramatic. We also reviewed the number of odd shapes in each group to examine effects of WNT upregulation. These shapes are often used as a measure of the presence of substances in the environment that are toxic to the planaria. The shapes we examined were normal, C-shapes, scrunched, and screw-like. The control group exhibited only normal shapes, while the LICL groups exhibited a majority of odd shapes after initial exposure. The fastest transformation from odd to normal shapes was in the 10 micromolar group in just 4 days.
This line graph shows the light-dark assay and the number of planaria that crossed through the dark region of the Petri dish in each group, which consisted of 4 head and 4 tail fragments. After crossing to the dark, planaria stayed in the dark without going back and forth to the light side. Most of the planaria that were in the 20 micromolar oppigenin group had a very similar number of planaria crossed through the dark region of the Petri dish as compared with the control group. The 10 micromolar group was not as good as the 20 micromolar, but much better than the LICL treated alone. In our study, LICL hampered regeneration and even caused worm death, but oppigenin was able to mitigate the effect. This was likely done with LICL upregulation WNT and then oppigenin reversing the effect at least to some extent. Regarding our regeneration assay, in the LICL to PEG group, planarian regeneration was hindered. In trials 1 and 2, the PEG group consistently lagged 2 stages behind control group, and its head shape never fully formed. In trial 3, the LICL to PEG group, all planaria died on the 3rd day they were observed. Planaria in the 20 micromolar oppigenin group regenerated the fastest compared with the other LICL groups. It caught up to the control group by day 6 in trial 1, by day 13 in trial 2, and by day 9 in trial 3. This variability was likely due to the planaria's sensitivity to even minute changes in concentration. The 10 micromolar oppigenin was also effective to an extent. It caught up to the control group by day 10 in trial 1 and by day 15 in trial 3. For the odd shapes assay in the 10 micromolar oppigenin group, there was the fastest transformation from odd to normal shapes in 4 days, compared to 6 days in LICL to PEG group and the 20 micromolar group.
Two measures of planarian behavior, PLMV and light-dark assay, both showed improved conditions of the LICL-treated planaria as a result of oppigenin treatment. On average, there was the fastest increase in velocity over time in the 20 micromolar oppigenin group. LICL-treated groups were much slower than control to start, and all LICL to oppigenin groups improved. Although there was no significant difference between the 20 micromolar group and the PEG group with a p-value of 0.122, it is of interest to see if the difference is significant with a larger sample size. We observed slight decreases in velocity in the 20 micromolar oppigenin groups after day 8. 20 micromolar was more effective, but also more toxic based on this measure. In the light-dark assay, oppigenin was effective in restoring darkness-seeking behavior. Both the 10 micromolar and 20 micromolar oppigenin groups had a higher number of planaria crossed to the dark region than the LICL to PEG-only group. Overall, our results were similar to those observed by Lin et al. In 2017 in colorectal cancer cells, which determined that oppigenin is effective at concentrations of 10 micromolar and over. Our study, which did not use cells but used planaria, showed the effectiveness of oppigenin in a whole organism rather than just cells. Major limitations we had were due to time constraints and limited equipment because of the hybrid schedule and work done at home. We also had a small sample size and had inconsistency of time intervals, temperatures, and locations. We would like to adjust our experimental setup in order to try to determine the optimal oppigenin concentration and time of exposure. Regarding future research, we would like to use pure oppigenin in place of the oppigenin that we used, which was supplemented with cellulose and not from a chemical supplier. We would like to test other oppigenin concentrations, including 15 micromolar and 30 micromolar. We would like to monitor odd shapes over longer time periods, as well as measure other indicators of planarian cognitive health, such as memory, and we would like to transfer planaria out of oppigenin after a fixed time interval.
To conclude, our study showed that LICL successfully hindered regeneration in planaria, likely through WNT upregulation, as well as hindered mobility and light-dark preference. Both the 10 micromolar and 20 micromolar oppigenin groups showed promising results. With the oppigenin group, we observed fully and correctly regenerated worms in contrast to the LICL-only treated worms. The oppigenin group also exhibited behavior similar to the control in 3 assays: mobility, normal rather than odd shapes, and light-dark preference. Although some toxicity of oppigenin was observed in the PLMV, 20 20 micromolar opicinin was most effective. Overall, our study indicates that opicinin should be studied further as a possible treatment for cancers involving WNT upregulation. Thank you so much for your time.